Asexual mode of reproduction. Let us study modes of reproduction involving a single parent without involving gametes. These are known as asexual modes of reproduction. Organisms can reproduce asexually in many ways. Some of them are given. Asexual mode of reproduction. Let us study modes of reproduction involving a single parent without involving gametes. Without involving gametes. These are known as asexual modes of reproduction. Organisms can reproduce asexually in many ways. Some of them are given here. Fission. Single-celled organisms such as paramecium and bacteria reproduce by splitting into two or more offsprings. This usually occurs in a symmetrical manner. They split into two by binary fission. When more cells are formed, it is called multiple fission. This is often the only mode of reproduction in these organisms. Budding. A growth on the body as a bud that grows to form nearly identical copy of parent. When the bud totally grows, then it separates from the parent and survives independently. Example, yeast. Fragmentation. Some can grow from a separate piece of parent organism. This can be from any part of the body. This happens only in the simplest, such as some flatworms, molds, lichens, spirogyra, grow in this manner. This may also reproduce sexually. Fragmentation is a common mode of reproduction in algae, fungi, and many land plants. Parthenogenesis. Nowadays, we are able to develop seedless fruits like watermelon, grapes. This is a process of reproduction where there is a shift from sexual to asexual mode of reproduction. This process also occurs in nature. An organism which reproduce sexually, sometimes asexually. We have utilized this process of reproduction in growing organisms of our choice with more desirable characters. In this process, generally the female gametes develops into zygote without fertilization. This stage kind of reproduction occur in best ants and wasps. The zygote might develop from fertilized egg or by parthenogenesis. When meiosis does occur, the parthenocarpic zygote develop into male monoploid, while the fertilized one developed into female diploid. Regeneration. Many organisms have the ability to give rise to new individual organisms from their body parts. That is, if the individual is somehow cut or broken up into many pieces, these pieces grow into separate individuals. This is similar to fragmentation. Vegetative propagation. In higher plants, vegetative propagation may be natural or artificial. Natural propagation. Leaves. In bryophyllum, small plants grow at the edge of leaves. Stems. Aerial weak stems like runners and stolons, when they touch the ground, give off adventitious roots. When the connection with the parent plants is broken, the stem portion with the adventitious roots develops into an independent plant. Examples for propagation by stem or from stolon, bulbs, cones, tuber, etc. Stolons, Vallisneria, strawberry. Bulbs, onion, cones. Tuber, potato. Roots of dahlia, radish, carrot, etc. grow as new plant. Artificial propagation. Cutting. Some plants grow individually when a piece of a parent plant having bud is cut from the existing plant. The lower part of this cutting is buried in moist soil. After few days, the cut parts having buds grow as an individual plant after developing roots. Example, rose. Layering. A branch of the plant with at least one node is bent towards the ground and a part of it is covered with moist soil, leaving the tip of the branch exposed above the ground. After some time, new roots develop from the part of the branch buried in the soil. The branch is then cut off from the parent plant. The part which has developed roots grows to become a new plant. Example, Nerium. Grafting. Two plants are joined together in such a way that two stems join and grow as a single plant, one which is attached to soil is called stalk, and the cut stem of another plant without roots is called scion. Both stalk and scion are tied with help of twine thread and covered by a polythene cover. Grafting is used to obtain a plant with desirable characters. This technique is very useful in propagating improved varieties of various flower and fruits. Example, mango, citrus, apple, rose. If you have two varieties of fruit yielding trees in your garden, one tree has the character of giving big sized fruits but less in number. The taste of the fruit is pretty good. The other one produce more number of fruits, but they are either big in size or tasty. By grafting a very young scion, that means a shoot part of a plant, can be made to flower and produce fruits quite fast. Collect information from your school library or internet about advantages and disadvantages of artificially vegetative propagation and discuss in our classroom. Spore formation. Generally, we may notice whitish threads and blackish powdery like substance on rotten fruits, bread slices, and other food materials. When you touch it, the blackish powder sticks on your fingers. These are the reproductive spores produced by a fungi, example, rhizopus. You have already learned about this in the chapter, The Story of Microorganisms, in class 8. Rhizopus produces hundreds of microscopic reproductive units called spores. When the spore case, it is also called sporangium. When the spore case bursts, the spore spreads into air. These airborne spores land on food or soil under favorable conditions like damp and warm conditions. They germinate and produce new individuals. Most of the fungi like Rhizopus, mucor, etc. Bacteria are non-flowering plants such as ferns and mosses reproduce by the method of spore formation. To examine rhizopus or common mold under the microscope, it is best to grow your own in a controlled environment. Use a soft bread that is preservative free or a roti. Fruits or vegetables such as potatoes or oranges. A good sample of mold may require 4 to 10 days to form spores, so be sure to plan ahead for this project. Leave the bread in the open air for about an hour so it is exposed to contaminants in the air. Place the bread in a plastic bag, sprinkle water over it to have dampness, then seal the bag, leaving some air inside. Place the bag in a dark, warm place. A kitchen cupboard close the stove may be one option. Or you could place it next to a window with a bowl or plate covering it from the light. Mold will grow best in a moist environment. Mold would start growing in 2 to 3 days, but will take a week or more to form spores depending upon the weather. Check the piece of bread every few days and add some water if it is drying. Avoid opening the plastic bag as much as you can. If you touch the bread, be sure to thoroughly wash your hands afterwards. When sufficient mold has formed, you can prepare a slide and examine it under the microscope. You would find whitish thread like growth with masses of black, gray, and green fine dotted structures. The black dotted structure is that of bread mold. Take a pot of the bread or roti to school in a matchbox and ask your teacher to help you to make a slide and observe under the microscope. Sporophyll. Ferns also produce spores. Collect a fern leaf, which is called sporophyll. Observe the leaf carefully. On the lower surface of the leaf, you find clusters of dot-like structures called sporangia. These contain spores. Gently rupture the sporangia with a needle and observe spores by using magnifying lens.